Sandhya, Assistant Professor of Emmanuel College of Pharmacy. Today we are going to discuss about the topic of morphology and ultrastructure of bacteria. Introduction. Bacteria are the first organisms to appear on Earth that thrives in diverse environments. This organism can live in a soil, the occasions, and inside the human gut. Humans' relationship with the bacteria is a complex. Some bacteria are harmful but more serve and useful of purpose. They support many forms of life, both plant and animal, and they are used in industrial and medicinal process. And repeat introduction. Bacteria are the first organism to appear on the earth. These organisms can live in a soil, the occasions, and inside the human gut. Humans' relationship with the bacteria is complex. Some bacteria are harmful, but most serve useful purposes. They support many forms of both plant and animal, and they are used in industrial and medicinal process. The general characteristics of bacteria. Bacteria are microscopic single-celled organisms. Bacteria are microscopic and single-celled organisms. They lack organelles such as chloroplasts and mitochondria, and they do not have the true nucleus. They lack organelles such as chloroplasts and mitochondria, and they do not have the true nucleus. Bacteria also have a cell membrane and a cell valve. Bacteria also have a cell membrane and a cell valve. The cell membrane and cell wall are referred to as the cell envelope. Asexual reproductions by binary fission. Asexual reproductions by binary fissions. However, some bacteria can also exchange genetic material among one another in a process known as a horizontal gene transfer. However, some bacteria can also exchange genetic material among one another in a process known as a horizontal gene transfer. Then next one, morphological of bacteria. Morphological of bacteria include shape, size, arrangement. Size and arrangement. Then first one, size of bacteria. Bacterial cells are around 1/10 the size of eukaryotic cells. Bacterial cells are about 1/10 the size of eukaryotic cells. Typically 0.5 to 5.0 micrometers in the length. Typically 0.5 to 5.0 micrometers in the length. Chromagnetic luminance is up to the half of millimeter long. Lithotripsine fish nosolis research 0.7 millimeter. Mycoplasma which measure only 0.3 micrometers. E. coli case 1.1 to 1.5 micrometer, while width 2.0 to 6.0 micrometer long. Sparnocatches 500 length. Cyclobacterium oscillosilius is about 7 diameter. Shapes of bacteria: coccus, chain, Spectrococcus, cluster, Spectrococcosis and Spectricloccosis. Bacillus, chain 1, Streptobacillus and Coccobacillus. Vibrio curved. Spiralium, Spirotium and square and square. Coccus, Bacillus, Coccobacillus, Vibrio, Spiralium, Spiralitus, square and star. This is shape of the bacteria. Array arrangement in bacteria. This is picture is available in focus. Then arrangement of the bases of the presence of flagella. Adequate substance of the flagella. Monotrichosis 1 flagella. Low photocratics. Tough at the one end. Ambiotartosis both ends. Petriocosis all around bacteria. All around pictures. The arrangement of the bases of presence of the flagella. Ultrastructure of bacteria. Flagella, pile and fimbulus, capsules and simple layers, cell wall, plasma membrane, mesosomes, cytoplasmic inclusions, neodymium and plasmids. Neodymium and plasmids. Flagella, pills and fimbulets, capsules and simple layers, cell wall, plasma membrane, mesosomes, cytoplastic inclusions, nucleodices and plasmids and nucleodices. Then ultrastructure of the bacteria. Ultrastructure of the bacteria. Flagella, pile and fimbulus, capsules. Posting flagella. Flagellum singular in a hair-like helical structure emerges from cell wall and cell membrane. It is responsible for motility of the bacteria. It is responsible for motility of bacteria. Size thin 50 to 20 nanometer in diameter. Single flagella can be seen with a light microscope only after staining with special staining, which increase the diameter of flagella. Non-cortical single protein subunit flagellum. Flagellum is a hair-like a helical structure from cell wall and cell membrane. It is responsible for mobility of the bacteria. Size thin 50 to 20 nanometers in the diameter. Single flagella can be seen with a light microscope only after training with a special training, which increases the diameter of the flagella. Non-cortical single protein subunit flagella. Then structure of flagella. Flagella has three parts: basical body, hook and fibulament. Basical body means it is a composition of the central rod in the to series of the rings, which is attached to cytoplasmic membrane and cell wall. L ring, it is a outer ring present only in a gram-negative bacteria. It is a lipopolysaccharide layer. L ring, it is the outer ring present only in the gram-negative bacteria. It is a lipopolysaccharide layer. Then P ring, it is a second ring in peptidoglycan layer of the cell valve. Peptidoglycan layer of the cell valve. M S ring in cytoplasmic membrane. First L ring for the gram-negative bacteria, lipopolysaccharide layer. P ring for peptidoglycan layer, cell valve. M and S ring is attached for the cytoplasmic membrane. Hooks means it is the wider region at the bases of ligament. It consists of filament to the motor protein in the bases. Length of the hook is longer in the gram-positive bacteria than gram-negative bacteria. Gram-positive bacteria than gram-negative bacteria. Filament, it is a thin hair-like structure rises from the hook. It is a thin hair-like structure rises from the hook. Structure of the flagella. Structure of the flagellus. Then first functions of the flagella, that acts primarily as an organizer of the locomotion's movement in the cells. They act as a sensory organ to detect temperature and pH changes. Few erythrocytes uses flagellum to increase reproduction rate. Recent research have provided that flagellas are also used as a secondary organ, for example, chlamydosis. that act as a primary as organ of the locomotion movement in the cells. They act as a sensory organ to detect temperature and pH changes. Few erythrocytes use spatulum to increase reproduction rates. Recent research have provided that flagella are also used as a secondary organs. File and fimbria. Both the fimbria and file are the hair-like appendants of the bacteria cell wall. They originate from cytoplasmic that produces outside after the penetrating the peptidoglycan layer of the cell wall. Familia are made up of 100% of protein called feminine sorpilions, which consist of about 163 amino acids. Smaller than flagella, adhere bacteria to surface. E. coli has numerous types. Then antibodies to well block adherence. F. filius is used to congestions. Exchange of generic information. Folitations increases. Folitations increases. That's of the fillets and fimbrans. Functions of filaments and filaments. Filaments have the adverse properties which attach the organism to the natural substrate or to the other organism. Filaments are regulated the blood cells such as erythrocytes, leukocytes, epithelial cells, etc. Filaments are equipped with antigenic properties as they act as thrombolysis and non-specific agglutinations. Family attaches the metabolic activity. Researchers make a contact between two cells since they possess how the act as the conjugations to capsules and simple layers. Some bacteria have an additional layer outside of the cell's wall called the glycosinosis. This coating of macromolecules protect the cell and help in adherence to surface. Simple layer glycoprotein molecules are loosely associated with the cell wall. Bacteria that are covered with the loose silent and protected from dehydrations and loss of nutrients. Capsules. The glycolysis is considered as a capsules when the polysaccharides are the more firmly attached to the cell wall. Capsules have the gummy strictly condensively and provide protections as well as the solid surface and to nutrient in the environment.